Okay, guys, this time I hope you can hear me. I really do, because this is the third time we're doing it. Uh, this is the second part of our option, microbes and the environment. The biggest part that we need to know for this is the nitrogen cycle. But before that, let me just briefly go over the roles of microbes in the environment. So you have producers, nitrogen fixers, and decomposers. Producers basically is just like plants. So these microbes, like cyanobacteria, they have what do you call it? They have chlorophyll, so they can photosynthesize. And basically, you know how photosynthesis happens. So I'm not gonna go into too much detail about that. The nitrogen fixation and nitrogen fixers I'll go over later because we'll talk about that in the nitrogen cycle. Decomposers. If you remember from our ecology unit, you remember that we talked about saprotrophs. So these saprophytic bacteria and fungi, they basically secrete hydrolytic enzymes so that they can digest the organic compounds. So as you can see, the producers convert inorganic materials like all the chemicals and stuff into organic materials that we can eat. Well, we don't really eat it, but other organisms do. And then as the plant material and other organic material dies and decays, you have decomposers who come in and release these inorganic nutrients back into the cycle. So this is like the plant cycle, basically. Nitrogen cycle. We have some very important parts and other not so important parts. In the book, we go over nitrification, nitrogen fixation, denitrification, and that's about it. If you look at the study guide, you can visualize everything a little bit more clearly. These two pictures, I have two after this one, I have another one. These two pictures are not as useful, but this is normally what you can see in an IB exam. You won't see all those clean diagrams. So get used to this. This is the second diagram. You can see how the animal and plant material are decomposed by the decomposers, and they're converted into inorganic materials like ammonia, which then is converted into nitrites and nitrates by organisms that I'll tell you about now. Those are the nitrification processes. Then what happens is all these nitrates go back into the plant, and then you can see how the denitrifying bacteria converts it back into the atmosphere, and the atmosphere comes back and gets fixated by legume root nodules, which I'll talk about. And then you have fertilizers that put it back into the soil. And atmospheric fixation, I'm not really so sure about, but I don't really think that's very important. You also have lightning and snow, I think that is, and rain that also help with the nitrogen cycle. So you can see that it's quite complex, but as I said before, if you look at the study guide, you'll find that it's pretty simple to follow if you have the basic stuff down. So let's go over the basic stuff. Nitrogen fixation. This is one of the most important parts. In this process, what happens is nitrogen gas and other nitrogen molecules are slapped onto carbon molecules, so you can create different things. Now, the big two bacteria that you need to know are azotobacteria and rhizobium. Azotobacteria, I like to remember by azonto. Get it? Yeah. So, azotobacteria and rhizobium are kind of different. Azotobacteria is not dependent on any plant. Rhizobium is a parasite that becomes mutually beneficial with leguminae. Azotobacteria, what they do is They're just free living. They can be anywhere and everywhere, and they just convert nitrogen gas into the other forms of nitrogen stuff. Uh, Most commonly ammonia. Rhizobium creates these nodules that you can see here, and leguminae. That's like legumes, if you know what I mean. So peas and wait, I have it somewhere. Uh, yeah. I suppose I don't have it. But basically, leguminae are like peas and all these plants with long roots. So you have the beans as well, peas and beans. Beans are the most common one. This is a picture from a bean plant. So one should be enough. If you need another one, you can find it. Yeah, you can also see that nitrogen fixation occurs during the Haber process, which I'll talk about later, and lightning. Then we have nitrification. Nitrification is when organisms like Nitrosomonas and Nitrobacter, which I don't have here. Nitrosomonas, what it does is it converts, why is this happening again? It converts ammonia into nitrite. This is important because it's nitrite, not rate, right. So, Nitrosomonas, what it does is it uses oxygen, so it's aerobic. So that's an important thing to remember. It uses oxygen to convert ammonia into nitrite, and Nitrobacter uses nitrite to convert to nitrate. Nitrates are very important. Nitrate are important for plant growth because they help in forming fertile soil, which is why farmers put fertilizer and stuff. Nitrification is favored by neutral pH, warmth, and well aerated soil. So if you see gardeners, they like to remove the topsoil and then mix it with the bottom soil because the bottom soil tends to have more nutrients, and that helps the plants. That's also keeping it arid and cool. Denitrification is the opposite. So useful nitrates are becoming nitrogen gas. One of the biggest ones you need to know is Pseudomonas denitrificans. Denitrificans is easy to remember. Pseudomonas. The other one was nitro. So remember that this is like the fake one. Pseudo, fake. Get it? So, denitrification would have the opposite, and it's important to complete the nitrogen cycle. So what it does is it converts nitrogen gas into nitrates into nitrogen gas, and that's bad for plant growth. It's favored in anaerobic conditions, so swamps and marshes and other waterlogged soils. Finally, we're at industrial fixation. So this is basically the Haber process, not Born-Haber cycle like we did in chemistry, but Haber process. It's basically this formula. So nitrogen and hydrogen, you get ammonia. Well, it's not really that important, so I won't spend too much time on it. Alright, so that's it for my part. I'll let Karan do the second part, which would be pollution of waterways and biomass for fuels and other solutions. See ya.